Welcome to this lecture on cell biology, where we will explore the fascinating world of the signal hypothesis. Imagine for a moment that you are running a massive, bustling Amazon fulfillment center. Every single second, thousands of different packages are being boxed up and placed onto conveyor belts. If these boxes did not have shipping labels, it would be absolute chaos, and no package would ever reach its correct destination. A biological cell faces the exact same logistical nightmare, acting like a microscopic factory producing millions of proteins every minute. Today, we will understand exactly how the cell solves this problem. Building on our fulfillment center analogy, let us look at the cellular sorting problem. Why does this matter? Because without a sorting mechanism, a cell simply cannot function. It produces millions of proteins every minute. Some of these proteins need to stay right where they are, floating in the main fluid of the cell. However, others need to be shipped outside the cell or delivered to highly specific compartments, like the endoplasmic reticulum. The cell needs a reliable way to tag these proteins with a biological shipping label so they end up in the exact right place. This brilliant mechanism of cellular sorting and tagging is exactly what the signal hypothesis explains. Now that we understand the problem, let us look at the solution discovered in the 1970s by Dr. Günter Blobel, a breakthrough so profound it earned him a Nobel Prize. He proposed that proteins destined for secretion, or for specific organelles, contain an intrinsic signal sequence built right into their structure. Think of this as the actual barcode on our package. This sequence is typically a short stretch of 15 to 30 highly hydrophobic amino acids. Crucially, this sequence is located at the very beginning of the protein chain. As the ribosome translates the messenger RNA and builds the protein, this signal sequence is the very first part to emerge, announcing the protein's destination to the rest of the cell. Since the signal sequence emerges first, the cell needs a way to read this barcode immediately. This brings us to a specialized molecule called the signal recognition particle. You can think of this particle as a cellular quality control inspector. It immediately spots the hydrophobic signal sequence as it emerges from the ribosome, binds to it, and temporarily pauses the entire translation process. Why pause? Because the protein is being built in the wrong location. The particle then acts like a microscopic tugboat, dragging the entire ribosome complex, along with the partially built protein, toward its correct destination. Following the journey of our cellular tugboat, we arrive at the destination, the membrane of the endoplasmic reticulum. Once the signal recognition particle docks the ribosome at this membrane, its job is done. The temporary pause is lifted and translation resumes. But now, instead of the protein being built into the open cellular fluid, the growing protein chain is pushed directly through a channel in the membrane and into the organelle. This elegant handoff ensures that the protein is safely compartmentalized exactly where it needs to be, ready for the next stage of its processing. Before we move on to the mathematics of this process, let us see this with a concrete visual diagram. Here, you can see the entire coordinated dance we just discussed. On the left, the ribosome is translating the messenger RNA, and the red signal sequence has just emerged. Notice how the signal recognition particle, labeled SRP, binds to it. Following the arrows, you see the entire complex docked onto the membrane of the endoplasmic reticulum. The translocon channel opens, and the growing protein is threaded inside. Finally, look at the bottom right. You will see an enzyme labeled signal peptidase acting like scissors to cut the red signal sequence off. This visual perfectly summarizes the physical mechanism. Now that we have visualized the mechanism, we must ask, why is this so critical to life? Understanding the signal hypothesis explains the fundamental organization of all complex eukaryotic cells. Without this mechanism, our cells would be completely unable to compartmentalize their functions. Imagine if highly destructive digestive enzymes were allowed to just float freely in the main cellular fluid. They would begin breaking down essential structures, potentially digesting the cell from the inside out. The signal sequence ensures these dangerous proteins are safely locked away inside the endoplasmic reticulum before they can do any harm. Beyond basic survival, this compartmentalization has massive real-world applications. This exact process is responsible for the secretion of vital hormones. For instance, without a signal sequence, insulin would never leave your pancreatic cells to regulate your blood sugar. Furthermore, many genetic diseases occur precisely because a mutation alters a protein signal sequence. When this biological zip code is broken, the protein gets lost inside the cell and cannot perform its job. By studying this system, scientists can now engineer synthetic proteins, tricking cells into mass-producing and secreting life-saving medicines directly into the bloodstream. This brings us to the final physical step before a protein is ready to function, which will lead us directly into our mathematical model. When the protein is pushed into the endoplasmic reticulum, it still has that biological shipping label attached, but the label is no longer needed. An enzyme called signal peptidase acts like molecular scissors and cuts the signal sequence off. This cleavage step is crucial because it leaves behind only the mature, functional protein. The length of this final protein depends entirely on how fast it was built, how long it was built for, and how much was cut off. Now that we understand the physical cleavage process, let us look at how we can represent this mathematically. The formula on screen calculates the final length of the mature protein. Lm represents this mature length, measured in the number of amino acids. This equals R, the rate of translation, multiplied by T, the total time in seconds. Multiplying rate by time gives us the total raw length synthesized by the ribosome. From this, we subtract S, which is the length of the signal sequence that gets cleaved off by the signal peptidase. If the translation rate R increases, the ribosome builds faster, resulting in a longer chain. Conversely, if the cleaved sequence S is unusually large, the final mature length, Lm, will decrease. Before we move on, let us see this formula in action. Imagine a pancreatic cell synthesizing a digestive enzyme. The messenger RNA has 401 codons. The signal sequence is 20 amino acids, and the translation rate is 5 amino acids per second. First, we find the total amino acids synthesized. Since the stop codon does not code for an amino acid, we subtract 1, giving 400 raw amino acids. Next, we find the mature length by subtracting the 20 amino acid signal sequence, leaving a functional enzyme of 380 amino acids. Finally, to find the total time, we divide the total synthesized length of 400 by the rate of 5. This gives exactly 80 seconds for the cell to fully translate this protein. Now that we have successfully used the formula under normal conditions, let us explore a slightly more complex scenario involving a cellular malfunction. Suppose a genetic mutation causes the signal peptidase enzyme to completely fail. A ribosome translates a new hormone at 4 amino acids per second for exactly 120 seconds. First, we calculate the total synthesized length by multiplying the rate of 4 by the time of 120, giving 480 amino acids. Next, we attempt to find the mature length. Because the molecular scissors are broken, the cleaved sequence S is exactly 0. Subtracting 0 leaves the final protein at 480 amino acids. Physically, this means the highly hydrophobic signal sequence is still attached, which will likely cause the protein to misfold or get permanently stuck in the membrane, rendering it useless and potentially toxic. We have covered a lot of ground today, so let us reinforce the most important ideas. First, remember that cells maintain order by using built-in sequences of amino acids as biological shipping labels for their proteins. Second, the signal recognition particle is the crucial quality control inspector that reads this label and guides the ribosome to the endoplasmic reticulum. Third, the signal peptidase enzyme acts as molecular scissors to remove the label, ensuring the protein folds correctly. Finally, we can mathematically model this process, proving that the mature protein length depends on the translation rate, total time, and the cleavage step. You now have a solid foundation in how cells communicate and maintain the delicate balance of life.